Hello, BISC130. This is the beginning of recorded lecture 2-3, starting off with chapter 7, cellular respiration. So, cellular respiration is the mother of all metabolic pathways. We talked about this in the last lecture, in the last chapter, a pathway to break or make complicated molecules. Cellular respiration is going to be a really, really important one that all living things have. So yeah, there's going to be a lot to this. Before we get to the pathway, though, there's one more thing I need to introduce regarding energy. So we talked about energy, again, in the last chapter, in the last recorded lecture. We talked about ATP carrying energy because of these covalent bonds. That, having covalent bonds means you have potential chemical energy, chemical potential energy, whatever. There's another way to have potential energy on the molecular, atomic, small stuff level, and that is through electrons. Electrons have potential energy, and this is the statement here, but it's a weird one to wrap your brain around. Yes, these electrons floating or buzzing around the nucleus of the atom, this figure from an earlier chapter, these things have potential energy just by existing. So an atom with a bunch of electrons has a bunch of energy, a bunch of potential energy in it. This also means if you have more electrons, you have more potential energy, and if you have fewer electrons, you have less potential energy. There's some terminology that comes with this, and here's a single slide that I think does a great job of laying this out. Here's our generic molecule A. If it loses an electron, it is going to have lower energy, color coded in yellow. A has lost an electron. It is now in a lower energy state. Electrons have energy. There is a name for this process. When an atom or molecule loses an electron or loses energy, this is called oxidation. And of course, there's the other side to this. Here is atom or molecule B. It gains an electron. When you gain an electron, you're going to gain energy. You're going to be at a higher energy level because electrons have energy. This process of gaining an electron and gaining energy is called reduction. And I see how that's confusing, and I do not make up these terms. I just present them. You're gaining electrons, but it's called reduction. You're gaining energy. You're going to a higher energy state, but it's called reduction. The reason why, there is a reason why, is because electrons are negatively charged. So if you are gaining electrons, even if you're gaining energy, your charge is going to become lower. It's going to become more negative. That's why this process is called reduction. So again, this is a great visual for this. Here's a summary. Electrons have potential energy. Losing electrons is called oxidation. You're at lower energy. Gaining of electrons is called reduction, and you're at a higher energy. You should really get familiar with these terms and know which one is which. And again, you could remember it's reduction because it's a lower charge, and then oxidation is the other one. But yeah, I'm going to be using these terms throughout this and the next chapter, so you really have to have these, got to get these solid in your head. And as this figure does imply, oftentimes if one atom or molecule is losing an electron, it's because it's giving it to another one. So this is a coupling of an oxidation and a reduction. One is getting oxidized, the other is getting reduced. So oxidations and reductions are oftentimes coupled, and we will see this. These reactions are called redox reactions, just reduction and oxidation. Redox reactions are when they're coupled together. Now, really what we're caring about in these redox reactions is the electrons, because that's the energy. It is worth noting, though, when we have redox reactions, oftentimes hydrogen ions will be transferred along with the electrons. So we're going to see examples of redox reactions, and we're going to see H plus just sort of pop out of nowhere. They're just transferred along with the electrons. So if you see H's appear as we do redox reactions, they're just coming along for the ride. Really, what we care about is the electrons. That's where the energy comes from. But I wanted to note this. We're going to see H is moving around too. So I'm bringing this up because throughout this chapter and the next chapter, also about a metabolic pathway, we are going to see some organic molecules that are able to easily be oxidized and reduced. So there are some organic molecules out there that readily gain electrons or lose electrons. They can become oxidized, they can become reduced. You can think of these things as little electron shuttles. They're called electron carriers, which is also a good name for these things. Little transportation trucks or whatever that are just going to take electrons from one part of the cell to another part of the cell, carry them, transfer them, whatever. An example, let me show you a visual, is something called nicotinamide adenine dinucleotide. No, you don't need to know that. It goes by the acronym NAD, but that does stand for something. Big complicated molecule here. But this is the positively charged oxidized form. I need to not mix these two up. This is the oxidized form. This is the reduced form. So this is the low energy form, and this is the higher energy form. This is the form that's ready to accept electrons. This is the form that has been given electrons. Empty, fully loaded. If you're trying to spot the difference here between one and the other, boom, here we go. It's right here in this part of the molecule. Again, you don't have to memorize the molecular structure or be able to draw these or anything, but if you're curious, the difference between the two forms is right up here. There's a single hydrogen here and a double bond in this ring here. The double bond is now a single bond, and an extra hydrogen. Like I said, sometimes H plus is transferred along with the electrons. That's why there's an extra H here. But again, what we care about is electrons have been given. So again, empty form, fully loaded form. We're going to see a couple of these in the next chapter. Just want you to be familiar with this concept. These things called electron carriers. To use our terminology, NAD plus is the oxidized form, NADH is the reduced form. Empty, fully loaded. They're going to go back and forth, shuttle their electrons around. One more thing about electrons before we can finally get into this metabolic pathway, and that is the idea that these electrons can exist at low energy states or at high energy states. And this is the most difficult thing to conceptualize regarding electrons and energy. I think the best way to try to show this is to think of these electrons as, again, they're orbiting a nucleus, and they can exist at a lower level, a lower energy state, closer to the nucleus. They can also exist at a higher level, at a higher energy state, farther from the nucleus. So we're going to see this in action. Again, this is just some background here, but you need to be comfortable with the idea of high energy electrons and low energy electrons. Because we're going to see high energy electrons, we're going to see low energy electrons, we're going to see them go from one state to the other. So you need to be just comfortable with this concept of electrons not all just being exactly the same. Some are high energy, some are low energy. It has to do with where they are in their orbit around the nucleus. So electrons can be a high or low energy state. All right, with that background out of the way, we can get into cellular respiration. So what is cellular respiration? Well, cellular respiration, double underlining this because there are going to be things underneath this, is the complete breakdown and oxidation of glucose to generate ATP. So we are going to take glucose, specifically this monosaccharide, this carbohydrate, this sugar. We are going to completely break down glucose. We are going to completely strip away all of its valuable electrons, and we are going to generate this very useful energy storage molecule, ATP. If we want to break down a sort of overview of this process, here's the equation. No, I have not balanced this equation. If you've taken chemistry, this is probably weird to you, but I don't care about balancing how many carbons on this side or whatever. This is mostly just to see the starting materials and the ending materials. So cellular respiration, starting materials over here, we start with C6H12O6. I don't like to make you memorize a bunch of chemical formulas, but you should know this one because it's really important. Glucose is C6H12O6. We start with glucose, obviously. We also have to start with O2. So we need oxygen to live. I think everyone knows that. We need to breathe. If we don't, we will die. We're going to see why this is true in cellular respiration. If we can't do cellular respiration, we're going to be dead pretty quickly. And that's because O2 is one of those required starting materials for cellular respiration. Okay, so we take glucose, we take oxygen gas, O2. Our ending products, by the time we finish this giant pathway, we are going to end up with CO2, which we just, we breathe out. This is a waste product. We get rid of this. We're also going to create some H2O, which is not easy for us to see. We're already giant bags of water, so we don't really notice the water being created by this process, but we will be creating some H2O. And we will be releasing some energy, which we could use to form ATP, energy in the form of ATP, which, as we discussed in the previous recorded lecture, is our go-to energy storage molecule short term to make stuff happen in the cell. So this is the big, as complicated as this gets, you can always come back to this and remember what the big picture is. We're breaking down, we're oxidizing glucose, we're trying to get ATP. That's the point of this whole thing. Okay, so how do we do this? Well, like I said, there are going to be several stages. That's why I double underlined cellular respiration. The first of these stages is something called glycolysis. So let's take a look at glycolysis. Oh, okay. Yeah, this is kind of maddening. Lucky for you, don't memorize this. But I did want to show this in order to just demonstrate what a metabolic pathway looks like. So this is a 10-step metabolic pathway. We're going to start with glucose and make glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-diphosphate, and yeah, in a series of 10 steps, with each step doing one small little thing to this, we're going to go from glucose to something called pyruvate. Every single step has an enzyme. Again, we're not memorizing the names of all these enzymes, but again, these are all known. This is what a metabolic pathway looks like. All these intermediates, all these enzymes. Mostly what I want you to know is what we start with, what we end with, and a couple of valuable things that happen in the middle. So this is a 10-enzyme metabolic pathway. We start with glucose, which we're counting the carbons here. Again, when we talk about organic molecules, we talk about how big they are by talking about how many carbons they have. Glucose is a 6-carbon molecule. By the time we finish, we are going to have pyruvate, which is a 3-carbon molecule. Actually, we're going to have 2 pyruvates, so we go from 6 to 3 plus 3. And yeah, if you're curious, there's pyruvate down here. 1 carbon, 2 carbon, 3 carbon. We're going to have a couple of those. So yeah, we haven't lost any carbons now. It took 10 enzymes to cut this thing in half. But it's more complicated than just cutting it in half. There are a few valuable things that happen along the way. If I can highlight those here. We got some ATP that we generate in this process. So if we look closely at this first step, we could see ATP to ADP. Wait a second. This isn't generating energy. This is spending energy. This is what energy consumption looks like. We're going from high energy triphosphate to this lower energy adenosine diphosphate. And we're doing the same thing here. This isn't generating energy. Well, this is generating energy. ADP goes to ATP. ADP goes to ATP. So we do have to spend some energy in a few of the earlier steps of glycolysis. But we are generating energy in the later steps of glycolysis. And I'm not going to, if you read the textbook, they will count exactly how many molecules of ATP happen in each step of cellular respiration. I'm not going to worry about that. But all of this stuff is happening X2, because this is where we split the 6 into 3 plus 3. So even though we spend some ATP in these steps, we are going to generate more than we have spent. So we generate a net amount of ATP. We gain a net amount of ATP. But yeah, we have to spend some initially in order to get this payout. So what's my summary here? ATP is spent. And any time in any chapter I talk about spending ATP, I'm talking about this chemical reaction, going from ATP to ADP in a loose phosphate that we've ripped off. So ATP is spent in some of the initial steps, but a net amount is generated. And again, whenever I say generate ATP, I'm talking not about building this thing from scratch. When I say ATP is generated, I'm talking about ADP and a loose phosphate, connect the two, create ATP from this. There's one other thing in this big pathway that I want to note, and I've circled it here. NAD to NADH. I told you these things would come up. Electron carriers. Remember, our big overview of cellular respiration, we are not just breaking down glucose, we are also oxidizing glucose. So that means we're removing valuable high energy electrons from glucose as we break it down. And yeah, that's what we see here. So NAD, remember, is the empty form of this electron carrier. NADH is the fully loaded high energy reduced form of this electron carrier. So we're oxidizing these intermediates and we are reducing NAD to NADH. So NAD plus reduced to NADH. This happens as part of glycolysis. And I should note, this part of cellular respiration, glycolysis, takes place in the cytoplasm of a eukaryotic cell. I'm pointing this out now because in eukaryotes, everything else after glycolysis is going to take place in the specialized organelles called mitochondria. So glycolysis is in the cytoplasm. In eukaryotes, which have mitochondria, all further steps are going to happen in mitochondria. So glycolysis in the cytoplasm, everything else in the mitochondria. Okay, so our next step here, we have pyruvate. We have two molecules of this, two of these 3 carbon pyruvate molecules. What comes next? Next is something called pyruvate oxidation. This is actually not a metabolic pathway. This step in cellular respiration is actually just a single chemical reaction, but it's a very important one. So we start with pyruvate, 1, 2, 3 carbons, and with a single enzyme, we're going to do a couple of things. We are going to, once again, rip off electrons from pyruvate. We're going to reduce NAD plus into NADH. So great, we're doing more oxidation of this carbon. We're getting more high energy electrons. The other thing that's going to happen in this chemical reaction is we're going to finally start ripping off carbons. We're going to take this 3 carbon pyruvate and we're going to rip off one of these carbons to create CO2. Remember, CO2 is one of our final products of cellular respiration. This is the end game for carbon. All our carbon in the initial glucose is going to be converted to CO2 by the time we're done with this. So if we're doing 3 minus 1, that leaves us with 2. We're going to end up with a 2 carbon molecule called an acetyl group. 1 carbon, 2 carbon. Now this 2 carbon thing is not just floating around as we do this reaction. We're going to take the remaining 2 carbons from pyruvate and attach them to something called CoA. And if you're racking your brain trying to think of the periodic table, like, is that cobalt, arsenic? No. This is not a chemical formula for some chemical compound. CoA is shorthand for coenzyme A, this monster. So, as I keep saying, don't memorize the exact chemical structure of this thing, but if you're curious, this is what coenzyme A looks like, and you can see why it's just shortened to CoA. For our purposes, this is just a carrier molecule. This is the whole job of CoA, is to hold on to these remaining 2 carbons and take them to the next part of their journey here. So, again, single slide, good summary of pyruvate oxidation. If I want to break this down into text, pyruvate oxidation. Pyruvate is broken down, oxidized, and then attached to coenzyme A, or CoA. This releases CO2. This generates some valuable NADH, and this results in acetyl-CoA, acetyl-CoA, the two remaining carbons from each of these pyruvates attached to the CoA carrier. Okay, so we have two of these. Again, we had two pyruvates at the end of glycolysis, so we're doing this twice. Now we have acetyl-CoA. We've got to deal with that. Our next stage in cellular respiration is something called the citric acid cycle. Let's get a visual on this. Oh, it's another one of these. Big metabolic pathway, a lot of individual intermediates, a lot of enzymes that aren't even labeled here, but there's a name for every single enzyme doing every single one of these reactions. Just like glycolysis, don't memorize this whole beast. This is a little different than glycolysis because it's not a straight line. Well, I guess our glycolysis figure was a little curved just so it fits on the screen. But yeah, glycolysis was a simple 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You end up something that is different from what you started with. This metabolic pathway, citric acid cycle, loops back on itself. It's a cycle, as the name implies. So again, we don't need to know every single step here, but the first one is very important. So this first step is going to take our acetyl-CoA from the last reaction. Yeah, there's our 2-carbon acetyl group attached to the CoA carrier. And for your convenience, I like this figure. It tells you how many carbons these have. So this says C2 underneath it, just keeping track. This has 2 carbons. We are going to take the acetyl group from CoA and attach it to something called oxaloacetate. Oxaloacetate is a 4-carbon molecule. 1, 2, 3, 4. So we're going to take 2 carbons, attach it to a 4-carbon. 2 plus 4 equals 6. The math is pretty simple here. And we are going to create a 6-carbon molecule called citrate. That's why this is called the citric acid cycle. The first step here involves in creating citrate. 1, 2, 3, 4, 5, 6. 6-carbon citrate. From there, citrate is going to be broken down. Hey, there's a carbon, CO2 being released. We're losing carbon. We're losing another carbon. And it's going to be oxidized. There are going to be more electrons removed from it until we eventually convert this citrate back to oxaloacetate, right where we started again, ready to receive another acetyl group and go through this whole business again. So let me start to summarize this. The citric acid cycle, we have an acetyl group, 2 carbons, is transferred from CoA to oxaloacetate, 4 carbons, to form citrate, 6 carbons. I know this is a lot of words here, oxaloacetate to citrate, but again, we're not memorizing any of these other intermediates here. These are, I think, the important ones to know. 2 plus 4 equals 6. Acetyl plus oxaloacetate equals citrate. Then, as I said, across several steps in this pathway, citrate is broken down and oxidized until it's back to oxaloacetate, and that completes our cycle. We're back right where we started. Now, a couple of CO2 is released. Again, these are the 2 carbons from the acetyl group we started with. CO2 X2 is released for each acetyl-CoA that comes into this. If you're keeping track of this, this is the end of the carbon. All the carbon we started with has finally been released into CO2. We started with 6 carbons. We went to 3 plus 3, 2 pyruvates. We lost a carbon from each pyruvate here in the oxidation. And here are the last 2 carbons from each pyruvate going to CO2 here. So by the time we finish the citric acid cycle twice, all of the carbons we started with in glucose are going to be released as CO2. So yep, CO2X2 is released for each acetyl-CoA. We also get some ATP that's generated. Now, technically this is GTP. Don't worry about it. GTP is functionally equivalent to ATP. I'm just going to say ATP generated to try to keep things simple. We are getting some ATP, some usable energy out of this citric acid cycle as a whole. And Of course, we're also oxidizing this, which means we get more of these high energy electron carriers. And here I've circled them: NAD plus to NADH, NAD plus to NADH, NAD plus to NADH, and FAD to FADH2. Yeah, it's another one of these. Flavin adenine dinucleotide is reduced to flavin adenine dinucleotide H2. Pause your screen now and spot the difference. Or not. Yeah, if you're curious, yep, there are the hydrogens that are passed along with these electrons. Again, we care about the electrons being passed. The hydrogens just sort of come along for the ride. But yeah, this is just another electron carrier carrying these valuable electrons. So in the citric acid cycle, yep, we generate some NADH, and which we've already talked about, and we're also going to have FAD reduced to FADH2. This is just another electron carrier, empty form, fully loaded form. So at this point, we have a lot of NADH from the previous steps as well, and we've got some FADH2 from this step. We got a lot of valuable electron carriers at this point as we finish out the citric acid cycle. Okay, so it may seem like we are done. I mean, as far as breakdown goes, we've completely broken down all the carbon we started with to CO2. Yeah, as far as breaking down glucose, we're completely finished. But this is not the end of cellular respiration. The point of cellular respiration was to get lots of ATP from glucose. We did get some ATP in the citric acid cycle. We did get some ATP in glycolysis, but most of the energy that we got from this breakdown and oxidation of glucose, most of that energy is in the form of these high energy electron carriers, NADH and FADH2. So we're not done with cellular respiration yet. We're about to start the trickiest part. What we have to do now is find a way to convert the energy of these high energy electrons into ATP. We're going to do that starting with something called the electron transport chain, or ETC. Let's get a visual on this. So if we want to look at what's going on in this electron transport chain, it looks like this. And there's a lot going on here, but we should see some familiar faces. We see NADH, we see FADH2, and look at what's happening to them here. NADH is going to NAD+, and FADH2 is going to FAD. These high energy reduced electron carriers are being oxidized back to their empty forms. They are giving away their valuable high energy electrons to this transport chain. That's what these black arrows are showing. This is what's going to happen to their electrons. So yeah, our carriers are losing their valuable electrons, going back to empty. What's happening to those electrons? Well, they're going to be transported from this protein to this molecule, to this protein, to this all these proteins have names that we're not memorizing, to this protein, to this protein. Again, it's a transport chain. Eventually, these electrons are going to be passed to O2 in order to create H2O. So this is why we need oxygen to live, because the final destination for these electrons from these electron carriers, as they're passed from protein to protein to protein to protein, their final destination is oxygen gas, which, again, don't worry about balancing this chemical formula here, but when you throw electrons onto oxygen gas, you're going to grab some hydrogens from the environment, and you're going to create H2O. So this is why we needed O2 as a starting material, and this is also why H2O was one of the ending materials. It's sort of a byproduct of this electron transport chain. But it's not just movement of these electrons. They're not just being passed from carrier to carrier to carrier. Their energy is also changing as they're passed from carrier to carrier to carrier. The best way to think about this is in a conceptual way. The electrons that we have on this NADH and FADH2, these are high energy electrons. As these are passed from these electron carriers to, this protein and this molecule and this protein, as they're passed from carrier to carrier to carrier in this transport chain, they lose some of their energy. They get lower energy. They lose more of their energy. They get lower energy until, again, final destination, oxygen gas, grab some hydrogens from the environment, from the surrounding water, create more H2O. So yes, we're going from electrons to an oxygen to H2O, but energy is lowering as we go. This is very important. So, big summary here. So in the electron transport chain, NADH and FADH2 are oxidized, again, just gotta keep using these terms, back to NAD plus and FAD. Their high energy electrons are passed through a series of other electron carriers. That's why it's called an electron transport chain. They lose energy with each transfer, so that by the time they are finally transferred to O2, they are low energy electrons, and of course this creates H2O. So this is the final journey of these electrons from these high energy electrons on the carriers all the way down to low energy electrons on H2O. But why? So we just lost a bunch of energy. We went from high energy to low energy. What was the point of this? Well, also remember the first law of thermodynamics, energy cannot be created or destroyed, but also, what do we do with this energy? What was the point of this? Well, we can see the point of this if we go back to our electron transport chain and look more closely at what's going on here. So we're passing electrons around. You may also notice this. Arrows pointing to this side of the membrane with a little H plus here. This is an example of active transport. You can't really see it too well on this figure, so I made this one to simplify it a bit. On the inside of this membrane, there are not a lot of hydrogen ions, protons, remember, same thing. There are a bunch of H plus on the other side of the membrane. We are using this energy of these electrons being passed, because again, these are high energy electrons going to lower energy electrons, moving down this staircase. That energy is not wasted. That energy is used to pump a proton from one side of the membrane to the other side of the membrane. It happens again here. Again, these electrons are going down their staircase. They're losing energy. The energy doesn't disappear. It's used to pump a proton. And again here, it's used to pump a proton. Again, this costs energy to do because there's already a bunch of it on one side, not much of it on the other side. This is, again, going back to a few chapters ago, this is active transport. So what this creates is a strong proton gradient. There are a bunch of protons, again, H plus, proton, hydrogen ion, all the same thing. Sorry if it's confusing, but it's just got a lot of names. Bunch of protons on one side, not a lot on the other side. That is a proton gradient. We have spent all of this energy from our food in order to create a proton gradient. In summary, energy from these electrons that are being passed along is used to pump protons across the membrane. This is active transport, and this creates a proton gradient. Once again, we have to ask, why though? What is the point of this? Why did we spend all of this valuable energy creating this setup? Well, first I want to go back to another slide from a previous chapter where we talked about diffusion across the membrane. And I want to point out that even though H plus is very small, H plus will not diffuse across this plasma membrane. It's charged. The lipid bilayer is hydrophobic. Any protons that we pump to this side, they're going to stay there. They're not going to be able to just zip through this lipid bilayer. So very important statement here, H plus cannot diffuse across the membrane. So we've created a situation where we have a bunch of protons on one side, not a lot of protons on the other side. These protons want to be on the bottom side. They want to equal out. That's just diffusion. That's like water flowing downhill. These protons want to be over here. They want the concentrations to be equal. This is a type of potential energy. This proton gradient is, again, an example of potential energy. You got a bunch of protons on one side that want to be on the other side, but they can't. They're being held back by this lipid bilayer. This is actually very similar to another example of potential energy that we saw in the last chapter, the dam and the waterfall. This water here in the reservoir wants to flow downhill, just like these protons want to flow from high concentration to lower concentration. But the water can't. The dam is in the way. It's holding it back. And these protons can't go where they want to go because this dam, this lipid bilayer, is in the way. It's preventing them from going the way they want to go. Now, this analogy is better than you may think because we have to think about why these dams are made. Why does this dam exist? Why was this constructed? What's the point? Well, here's a slide that I bet no one expected to see in an intro biology class. Here's how a hydroelectric dam works. So the reason why we build dams is there's a ton of potential energy up here in the reservoir. It wants to flow downhill, and we're going to let it. We're going to let it flow downhill, but as it flows downhill, it's going to spin a turbine, which is going to generate electricity. So there's a lot of this hydroelectric dam is converting potential energy of this water into electrical energy. So what we need is a way to convert this potential energy into some sort of usable energy. And just like the water flowing through the dam powers the spinning of a turbine, we're actually going to do a very similar thing in our cells using a protein. The protein that we're going to use to harness the energy of this proton gradient is a protein called ATP synthase. And what ATP synthase is going to do is it is going to allow these hydrogen ions, these H pluses, these protons, to flow through itself. ATP synthase spans the membrane. It's open on one end, it's open on the other end. And as these protons move through this big old protein complex, it is going to power the rotation of a stalk, a spin, that is going to generate ATP. And this is the figure from the same textbook I've been using, very familiar looking bilayer here, and all of these figures. This is, I don't like this because it makes ATP look too simple. Again, it's just a blue blob. ATP synthase is actually one of the most sophisticated, complicated enzymes in all of biology. It looks really cool. And so here's a different textbook's, version of this. It's still blobby cartoons, but you could see, there's a rotor here. There's a stalk. Part of this actually spins. This is a little confusing because this textbook is sort of drawn upside down from this one. The figures I've been using show a bunch of protons on this side and not a lot of protons on the bottom side. This one is just showing it in the upside down way. There are a bunch of protons on this side of the membrane, not a lot of protons on this side of the membrane. But yeah, they're going to pass through ATP synthase. It's hollow, it's big. That movement, that rush, that flow of protons is going to cause this stalk to spin. just like the movement of water causes this turbine to spin, that physical movement is going to power the creation of ATP from ADP and phosphate. So, whoa, this is a lot to take in. Let me try to summarize this. But again, these visuals are great. Let me try to put this in words, though. So our last stage here is named after the enzyme. So we go from the electron transport chain. Our final step here, I'm just calling ATP synthase. This is a large multi-protein complex. Again, to use a term from several chapters ago, this has a quaternary structure. It's a bunch of different proteins that come together to form this big thing. It spans the membrane, again, reaches from one side to the other. It allows protons to pass through it down their concentration gradient, just like flowing downhill from high concentration to low concentration. It allows them to flow where they want to go. This powers the rotation of a stalk, which generates ATP. Importantly, this needs a proton gradient in order to work. If there was no water in the reservoir, the dam would not be able to generate any electricity. And if we did not have a bunch of protons on one side and not a lot on the other side, ATP synthase wouldn't be able to do anything either. So I presented the electron transport chain and then ATP synthase, but really, in the mitochondria, they're right next to one another. They're working at the same time. So here's citric acid cycle, creating NADH and FADH2. Here's our electron transport chain, working hard to pump protons from one side of the membrane to the other. And then right next door is ATP synthase, using this proton gradient, allowing these protons to flow from one side of the membrane back to this side and generate ATP. So all of these things are incredibly important, and they work together. In fact, again, the textbook will count exactly how many molecules of ATP are made at each stage along the way. Don't worry about that. I'm just going to say, in sort of qualitative terms, most of our ATP comes right here. We did get some from glycolysis and the citric acid cycle, but the biggest payout, most of the ATP from all of cellular respiration is made by ATP synthase, is made at this stage. And that means anything that shuts this down, anything that shuts this down is going to be devastating. I think in the last chapter we were talking about enzyme inhibitors. Arsenic and cyanide are both enzyme inhibitors that shut down these things. They shut down different parts of the electron transport chain. And cyanide will kill you real good, because if you can't do the electron transport chain, you can't pump protons, which means there's going to be no proton gradient, which means ATP synthase can't do its job. Most of our ATP comes from ATP synthase, so you're going to be dead due to lack of energy, due to lack of ATP. So yes, these two are right next to one another, and this is our big payoff of everything. Most of our ATP comes from ATP synthase. Okay, so one final thing in this chapter. All of this whole pathway, all of cellular respiration was about glucose. We started with glucose, and we ended up with water and ATP. Glucose, obviously a very important source of energy, obviously a sugar that we use across the body in many different cells, but glucose is not the only source of energy we have in our body. What about all the other stuff? What about all the other carbohydrates? What about lipids and fats? What about amino acids, proteins? We get energy from proteins that we eat in our food. What about all the other stuff? Well, lucky for us, all these other sources of energy from our food do not have their own, completely separate pathway. They're all going to funnel into cellular respiration. So let me break this down. Let's start by talking about other carbs. So what about the catabolism, again, breakdown pathway? What about the catabolism of other carbohydrates? Stuff like big polysaccharides or other monosaccharides or disaccharides. How do we get energy out of these things? Well, it's real simple. They're broken to monosaccharides, and then they go to glycolysis. So yeah, this is pretty easy. If you want to get energy out of any other carb, you just turn it into a monosaccharide, send it into glycolysis, and then business as usual. Go through the whole rest of the pathway as normal. That's easy. What about proteins? Well, proteins, if you'll remember this slide, are a string of amino acids. Here's our primary structure here. It's just beads on a string. It's just a bunch of amino acids connected together. If you want to get energy out of a protein, you start by snipping the protein down into its individual amino acids. And so you're going to end up with a pool of a bunch of different amino acids. From there, we just shuttle them into somewhere in cellular respiration. Again, this looks crazy, but honestly, this should look familiar. Here is glycolysis, glucose to pyruvate. There's pyruvate oxidation, and here's the citric acid cycle. Don't memorize all of these, but this is just showing all those amino acids are able to enter at some point, depending on how big they are. So some of them hop in here in the acetyl step. Some of them hop in here, replacing pyruvate in this overall pathway. Some of them jump in midway through the citric acid cycle. But yeah, they could all be dealt with one way or another. You just need to break them down to amino acids first, and then they're going to enter this pathway somewhere. So catabolism of proteins. Proteins are broken to amino acids, where they will either enter glycolysis, pyruvate oxidation, or the citric acid cycle, and yeah, they'll be broken down completely to CO2 one way or another. So we can get energy out of proteins in this way. What about our third source of energy from food? What about fat? What about lipids and fatty acids? These things like this triglyceride. Well, if we want to get energy out of this, we are going to take the fatty acid tails and we are going to snip them into 2 carbon units. From there, we can send them to the citric acid cycle, which starts with this 2-carbon acetyl group, and it's going to be business as usual from there. So again, we're not inventing some brand new pathway to deal with these different sources of energy, these things other than glucose. We're just sending them into the same pathway we have. They just enter at a slightly different point. So lipids and fatty acids broken into 2 carbon units, and then they enter the citric acid cycle, and business as usual. Here's an overview of all of this. Carbohydrates and some amino acids go to glycolysis. Some amino acids come in here at the pyruvate oxidation step. Fatty acids and some amino acids come in here at the citric acid cycle, but it's all going to eventually lead to, this says oxidative phosphorylation. That's not a term we talked about. This means the electron transport chain and then ATP synthase. So they're all going to get finished off one way or another. Okay, so that takes us to the end of cellular respiration, the end of recorded lecture 2-3. Maybe I should have mentioned this at the beginning, but in retrospect, I do want to point out, this might be the most tricky subject in the entire quarter. This might be the most complex topic, either this or the next chapter, in the entire course. So in this way, you're lucky that we have these recorded lectures. If there's any part of this that you're hung up on, that doesn't make sense, you're free to rewind, skip back to another part of the video, watch this again, and certainly, as with every lecture, feel free to email me. Come visit me during my my office hours if you want to. This is, I understand, a pretty tricky topic. So there's another tricky topic coming up in the next recorded lecture. But yes, this is the end of cellular respiration. This is the end of recorded lecture 2-3.